send me a text that he's just arriving to the consultory. Okay? It's okay.
Okay, perfect. I'm gonna start sharing my screen then. Okay. So I hope you can all see it. Okay, cool, perfect. So this is an informative session, and we're gonna discuss a little bit about the quality controls that we have in the lab, where we culture the mesenchymal stem cells. My name is Mariana Orozco, and I'm the chief of the quality control department, and also collaborate in immunotherapy and also in the culture of the cells. I have a master's degree in cell biology. And well, I'm going to start giving you a brief introduction about what the mesenchymal stem cells are. And this is a, let me do this. This is a photography, a micro photography of electron microscopy of how the mesenchymal stem cells look. And all of these things that you see on top of the cells are vesicles that the cells are releasing into the media. And this is how you can see them by like through fluorescence microscopy. The green dots is the nucleus of the cells, and the rest is the cytoplasm of the cells. So in order to a mesenchymal stem cell to be considered as one, it has to meet some criteria, okay? One of them, like I show you here, this is not like precisely the order of importance, but one characteristic is that these cells are adherent to plastic, and this is how we can like differentiate them among other populations of cell populations that we can find in the tissue that we're going to obtain these cells from. For example, if we get these cells in a suspension as blood, the ones that are going to be attached to the plastic in the way that it's a flask, of a culture flask, these are probably or most likely going to be the mesenchymal stem cells. The second criteria is that these cells have to present certain markers or certain molecules in their surface or in the cell membrane, and we can differentiate these cells from other cell populations because of the expression of these markers. I'm going to talk more a little bit of this in the presentation, but there are some molecules that we're going to look for the expression in these cells, and depending on their positive expression or negative expression, we can exclude or include the cells into this population. And the other feature, which for me is the most important one, is that these cells have a multipotent differentiation potential. This means that these cells, they're going to have the ability to self-renew themselves. So they're going to have, like, they're going to proliferate, and they're going to make a pool of other mesenchymal stem cells. And from these ones, according to the signals that we're going to get from the environment, or basically the growth factors that are in the environment, the signal that these mesenchymal stem cells are going to receive, they can differentiate in different types of tissues or different type of cells, okay? And speaking of this, we can obtain the mesenchymal stem cells from different tissues. We can get them from bone marrow, from adipose tissue, from dental pulp, and also from umbilical cord and placenta, which is the ones that we're gonna concentrate and focus on, because these are the ones that we use, or these tissues are the ones that we use to get our mesenchymal stem cells. So as I said, these mesenchymal stem cells, they have the ability to self-renew themselves and also to differentiate in other cell types. more specifically, they're going to differentiate in connective tissue, cartilage, adipose tissue, and bone tissue, okay? But also their interaction at the release of growth factors to the media, they can have the effect of activate or give the signals to other cells, such as epithelial cells or neuron cells, to also regenerate and proliferate, okay? Because of these cell growth factors that these mesenchymal stem cells can release into the media, they go to other type of tissues, such as muscle as well, lung cells, etc. And they can have this effect of make them regenerate as well. Not only the ones that come from the mesoderm, which is the germinal layer, embryonic germinal layer that they come from, but also other tissues, such as neurons, which is very important for this talk, obviously. And according to this, I just did like a quick search on Google to see what is the relationship between mesenchymal stem cell therapy and treating autism. So this is a research paper from 2014, and one of the highlights of this research paper is that, well, it's, sorry, as you can read here, is putting these mesenchymal stem cells as a promising candidate to be a therapeutic option to the treatment of autism. Okay, this was 2014. And I think this one is from 2020 probably. We have this other research paper. They use human umbilical cord mesenchymal stem cells to treat ASD, autism spectrum disorder in children. And they talk about the safety profile and the effect on cytokine levels. Basically, what it says here, it is the effect or the effect that these cells are having is to reduce inflammation or like an inflammatory associated neurological condition. And these children they use with ASD, okay, to perform this essay that comes from this research paper. And basically what it says is that repeated infusions of these mesenchymal stem cells are safe and resulting only in mild or moderate adverse events, which probably are like just a little bit of discomfort, headache. They have not serious adverse events related to the treatment. And also, these injections help to reduce the levels of inflammatory cytokines that were detected in these children. And another example is this one that is a research paper. It was being performed in mice, and it's a model of ASD or autism. In mice, they give these mice valproic acid to make them have this autism-like behavior. In the female, in the female mice, they are pregnant. So the offspring of these treated mice, they're gonna be born with this autism-like behavior. And what they did is to take the umbilical cord, the human umbilical cord mesenchymal stem cells exosomes, and treat these mice with these exosomes by an intranasal injection. And apparently, well, this reduced or had an anti-inflammatory effect on the mice and helped to improve this autism-like behavior. So in this case, or in the previous one, they use cells, and in this one, they use exosomes, okay? Another concern that I've been asked many times is, and that I want to make this very clear, is that this cell therapy or the treatment with cells, the mesenchymal stem cells, it doesn't mean that we're performing a gene therapy, okay? So these cells are gonna have their own like DNA that obviously comes from the donor that we're getting these stem cells, but that doesn't mean that these genes that come in these cells are gonna affect our one or our DNA, or they're gonna mix with our one, and they're gonna have an effect in that matter. What I just explained to you is that these mesenchymal stem cells have an effect, like attaching to the tissue that needs to be repaired, and also releasing growth factors into the environment. And these factors are going to activate the cells from our own body and to have this effect of regeneration of the tissue, or anti-inflammatory, or releasing or anti-inflammatory cytokines, or decreasing the levels of inflammatory cytokines due to this immunomodulatory effect that they can have in the immune system. The interaction of these mesenchymal stem cells with cells from the immune system, such as T cells or B cells or NK cells or neutrophils, this can have at the end this outcome that it might be an anti-inflammatory effect or an immunomodulatory effect that we see. But this doesn't mean that it's in any way, shape, or form changing our own DNA and make it like differently or mix it with the ones that comes from the cells. That's completely false, okay? And I just wanted to make that very clear. Another thing that I want to tell you is how do we select our donors? And basically we select healthy young women that are in their 37 from 38 weeks of gestation, and which birth is going to be through C-section or cesarean section. This to avoid the contamination through the big channel of the tissue that we're going to get, that is the placenta. And obviously we're going to have the record of each donor, and we're going to make sure that these ones, like obviously they're like healthy and they don't have any infections or any diseases or pathologies or inflammatory, degenerative pathologies, etc. We don't, we exclude those ones from the donors that we need, and also they have to be negative from this lab test, basically for syphilis, hepatitis B, C, HIV, among others. Okay, so we want these donors to be healthy, young women, younger than 30 years old, and that's how we select the tissue, to have like at the beginning to start with, to have very good quality of the tissue that we're gonna start with. And after we get the tissue, it can be the placenta or the umbilical cord that we obtain the Wharton jelly from. We're gonna process the sample, and we're gonna collect the mesenchymal stem cells from these samples, and we're gonna maintain these ones in cell culture with certain specific conditions of oxygen, carbon dioxide, humidity, etc., temperature, also to maintain these cells in the best way possible. And we're going to obtain one sample of these cells that we start in culturing, and the people from this like production area, they're going to send it to me so I can analyze these cells to check if they meet the criteria, as I told you in the first slide, to meet if these cells meet the criteria of being mesenchymal stem cells. So what I do is just check through flow cytometry, which is going to explain to you what that is, to check whether these cells are or not the stem cells that we're looking for. And after that, I can say like, okay, these cells are okay, and you can use them to give, to make doses and put them into vials and deliver it to the patients like at the end. So which each one of the doses that we deliver, we are making, we have our quality control certificate, and it looks like this one. So in the top part of the certificate, sorry, we have the date which they are produced, the batch, or this batch is related to the cell line that we're culturing, and we have different sections of this certificate. The first one, it's telling us the morphology, how the cells, the mesenchymal stem cells, look like in culture, how viable they are. That means like how long they can last in a physiological solution after like being like taking or harvest from the culture and putting into a physiological solution to make into a dose, how long they're going to last alive. And the other section is what about their surface markers they're expressing. So we can have, or the cell lines, they can have variations between the expression of the markers. We can get this variation from cell line to cell line. We know, or I presented to you, that these markers, in order to be considered as positive, it needs to be an expression of more than 95%. And to be considered negative, they have to have an expression of less than 2% in the sample that we are analyzing. I'm gonna get to it more. And the next section is about the oncogenes. And the last one is the biological analysis. And this means which, if the sample that we are analyzing, it has some kind of pollution with some contaminant or bacteria or fungi or etc. Obviously, we don't want that in our cells that we're going to inject into the patients. And this slide is to show you how these mesenchymal stem cells look like in culture. These ones come from placenta, and these ones from Wharton jelly. They do like present like differences on how they look in culture. The stem cells from placenta, they look a little bit more like that they have a fibroblast morphology, and the ones from umbilical cord, they tend to grow like more out of place, like from, I don't know how to explain this to you, but they do grow like differently, and you can differentiate them in culture because they are going to have like different characteristics, at least in the way that they look. But as you can see, or as you're gonna see in the other slides, like they do present these characteristics or these molecules in their surface, and we can consider them as mesenchymal stem cells. As I told you, the mesenchymal stem cells from adipose tissue, from dental pulp, or from placenta, they do have to present these molecules to be considered as mesenchymal stem cells, but also it is very important to check and to monitor every day how this mesenchymal stem cell look in culture, because this is one way to see whether they're growing adequately, or if they have any contaminant, or if they are being submitted to lots of stress, or I don't know, other things that we need to evaluate to make sure that these cells are in the best conditions possible. And I explained to you that we have in the certificate a method to check the viability of these cells, and this means is the percentage of the proportion of live cells that we have in our sample. We use just a very quick assay to tell us how many, or how's the percentage of these alive cells from the total of the sample. And what we use, it's a dye, a viability dye. So what it does is just to exclude the cells that are viable. So this colorant, in this case, are these things like green green molecules that are represented here, they cannot go into the live cells because they have their full integrity of the membrane, of the cell membrane. But the ones that are apoptotic, which means they are in process of dying, or the dead ones, they're gonna let the the green ones are some molecules that are expressed in the live cells, and the red dots are the ones that are the colorant, that died. So the ones that are dying or they are dead, they're gonna let the colorant go into the cytoplasm and nucleus, and this is how are we going to differentiate the ones that are alive from the dead ones. And this is what they did here. It was a kinetic of the viability, and this lab, what they did, it was taking one of our samples of cells and to analyze them every 24 hours, from 24, 48, 120, etc., to check how much of these cells were dead after this time. And as you can see here, from the basically the first 4 days, the cells remain highly viable. More than 98% of the cells in the sample remain alive until after 96 hours of being harvested. And this means that our cells, they can remain very, very, like in very good conditions, even after 4 days of being taken out of the culture conditions. And this is what we have, this expiration date that you can have, or you can check in the bio that you're gonna get from the doses. So this L001 is the batch that is related to the certificate that you're gonna get. And this CAD, which is the Spanish for expiration date, it tells us like how many days they're gonna have to be alive, the cells. Like in this case, and according to the previous results that I showed you, we decided to give the cells an expiration date of 3 days. Okay, and you can check in every bio. This, the batch and the number is the date which the cells were produced, and the expiration date is 3 days after. So we advise you to use these cells after, obviously, if they have been preserved in optimal temperature, which is from 2 to 8 degrees, and it has less than 3 days, you can safely use them without like expecting to having any problem because of this. And I mentioned to you something about flow cytometry. And flow cytometry, it's a technique which is like extensively used in science, specifically like in cell biology, to analyze cells. Cells that are in a cell suspension, which is in a liquid, and we can use this machine that is called flow cytometer. And this machine has several lasers, and the specific one, it has 3 lasers, but there are very big machines that can have like up to 6 lasers. And these lasers, what they're going to do is to go through every cell that is in the cell suspension, and according to the light that they're gonna, like each cell are gonna get this beam of light that is coming from the laser, and according to the light that is going to be scattered from this cell is the signal that the machine is going to get, and it's going to give us a digital signal that we can interpret as being like positive or not to the molecule that we're looking for. Okay, in this case, what we use is antibodies, and these antibodies are attached to a fluorochrome, or to a fluorescent molecule. And if these molecules have been activated by these lasers, we are going to obtain this signal that we can interpret and we're going to see like how many of these cells, or what is the proportion of these cells that are positive in the sample for the molecule that we're looking for, and how many molecules of this thing that we're looking for in the cells it's present in each of these cells that we are analyzing in the population. I know that this sounds like quite technical, but it's actually very easy to manipulate once you have the experience. So as I told you, mesenchymal stem cells, they have like different surface markers that they need to express to be considered mesenchymal stem cells. Some of them are these ones that are in red, in this positive column, that as I told you, each sample has to have more than 95% of positivity for these molecules: CD44, CD90, CD105, CD73, and CD146, among others. But also, on the other hand, they have to be negative, or they don't have to have other markers such as CD34, CD45, CD31, HLR-DR, and other molecules that are present in immune cells. Why is this important, and why am I telling you this? It might not mean anything to you, talking about CD44, CD blah, 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 but these molecules, or there are receptors that the mesenchymal stem cells, they have in their membrane, and they're important to their function. Some of them, like these CD73, they are enzymes, or they act as enzymes. CD90, and it's a like anchorage molecule, or they help the cells to attach to other surfaces, in this case, like to other cells, or to interact with other cells, or also with the surface they are attached to, as I told you, like plastic. CD44 is the receptor of hyaluronic acid, which gives the signals to the cells to migrate and to adhere also to other cells or to surfaces. And CD105, it's a glycoprotein that is a member of the TGF family. And this TGF beta, it's very important because it's an anti-inflammatory cytokine that is present in our body. And it is important because it tells the body to reduce inflammation. So this is, it might be one of the reasons why these mesenchymal stem cells, they have this effect when they're injected in the patients, because of like, they have the ability to interact with this cytokine, or even release these kind of factors into the environment. And on the other hand, these mesenchymal stem cells should now have the expression of molecules that are present in hematopoietic progenitors, such as CD34, and they don't have to have it because they are not hematopoietic cells. And also, they don't have to have a CD45, for example, because there are no leukocytes. And these, if we have samples that they have more than 2% of these markers, it means that our sample is contaminated with other cell populations that we don't want, and we cannot consider this as a mesenchymal stem cell sample, okay? And it is important also not to have HLA-DR, because this molecule is the one that the cells from the immune system are going to recognize as a signal, and they can be rejected because of this. So these mesenchymal stem cells, they have a very low expression of these histocompatibility molecules, and this is why they are not rejected when they are injected into another patient, even when it's not an autologous injection, okay? And I wanted to show you this. This is these histograms are the analysis of one sample of our cells from mesenchymal stem cells. And as you can see here, the blue ones are our sample, the gray one is the control, And you can see there are samples expressing these molecules as surface markers from mesenchymal stem cells that we have, that we want to have, the CD44, CD73, CD90, CD105, and they lack the expression of the ones that we don't want, basically, or they're negative to CD34, CD45, and the MHC2 molecules. And this is an example from a research paper that I took, where you can see in fluorescence how these cells present the expression of these markers. You can see here like graphically, and they lack the expression of the ones that we don't want, basically. Another important thing about that we need to analyze in the mesenchymal stem cells is how if they have the expression of something that is called oncogenes. Oncogenes, they are normal genes that they have been mutated, or which expression is abnormal compared to the normal expression of the other cells. Okay, these oncogenes are proteins, or are genes that control the expression of certain proteins, which are they're going to control the cell cycle of these mesenchymal stem cells. This means that these molecules are going to tell the cells when to divide, or when to proliferate, and when to stop. Okay, if these oncogenes are mutated, these can lead to the appearance of certain type of cancer or certain type of leukemia. And as you can see here in this drawing here, is the different oncogenes and how important they are, or how much they are expressed in different type of cancer. For example, lung cancer, breast cancer, liver, stomach, colon, etc. So it is very important to us to analyze our cells, because as I told you in the beginning, the mesenchymal stem cells have the ability to proliferate, and they do proliferate loads, and they renew themselves. So we have to make sure that our cells, they do have their checkpoints in order. Okay, that's why we check, or we perform this oncogenicity analysis, and we check 4 different, 3 oncogenes and 2 more, the P53, that is this guardian of the genome that basically monitors genetic stability. So P53 we check c-MYC, p-MYC, and CRAS. So basically these 3 of here that I'm signaling now, these ones are oncogenes that tell the cells when to divide. Okay, so it's normal, the expression in the sample from mesenchymal stem cell is normal. When on the other hand, P53 is a protein that tells the cells when to stop dividing. So it is very important to have this one, because it's like a brake of a car telling when to stop. Okay, it is important to have this one in order as well. So we have an important expression of this P53, which is very good. And we have just a background expression of the oncogenes, which is very important, and it's telling us that our cells that don't present any mutations in these molecules that monitor the cell proliferation and DNA stability. Also, we have the analysis from the microbiology telling us that our cells are not contaminated with either bacteria, fungi, yeast, etc. Okay, this is very easy. I'm not going to stop much in here. And another thing that I want to tell you is about the exosomes derived from mesenchymal stem cells. This diagram is showing us a cell, mesenchymal stem cells, And this one, this arrow, comes from these very tiny small vesicles that the mesenchymal stem cells are releasing into the media. And these exosomes, they have different content of a mix of content of proteins, DNA, RNA, microRNAs, that are basically information to the other cells or to the environment, and sending signals to the other ones to tell them like what to do or what is going on in the environment. So these exosomes, like you can see here in this electron microscopy image, these arrows, these blue arrows, are marking the exosomes. So as you can see how tiny they are like 100 times smaller than the cells. Because if you imagine this being a cell, is like, it goes like all the way like out to the screen, if you can see it here, like very, very big. And it's just tiny, tiny vesicles that have this, that contain DNA, proteins, enzymes that are sending to the environment, as you can see here as well. And the red arrows, how tiny and how small these exosomes are. And this represents a big advantage, like compared to the cells, because they are smaller to the cells. They have low immunogenicity as well, because they have a low expression of the HLA molecules as well, and the delivery. These exosomes are highly unlikely that they're going to get trapped in lungs or other tissues that can happen to the mesenchymal stem cells. So if we want to improve the delivery of the effect of the mesenchymal stem cells, exosomes the exosomes are a very good alternative to this, for this effect. And the exosomes have their certificate as well. You have a photograph of these exosomes that come from the mesenchymal stem cells. You can see it here, and like I told you, they're like 100 times smaller than the stem cells. And they have, we checked the expression of CD9 and CD63, which maybe you like missed it here, but are molecules or membrane proteins that are expressed in these exosomes that help us to differentiate these exosomes from other type of vesicles, of secreted vesicles from the cells. So they have the expression of CD9, CD63, CD81, and CD82. And this is how we differentiate them from other type of vesicles. That's why we analyze them here, to make sure that the ones that we are obtaining are actually exosomes. And also the content. I showed you that they can have proteins, exosomes, DNA, RNA, and basically this translates to information to the other cells. And this is just an example, like how or in what kind of conditions exosomes have been used because of their effect, preventing apoptosis, oxidative stress, fibrosis, inflammation, okay, and promoting other type of proliferation, differentiation, and angiogenesis. And these is being used to treat different conditions, as I told you, and these include brain damage, brain injury, ischemic brain injury, autism, and that's why I put it here. And I'm just getting to the end. At the concluding remarks is that the mesenchymal stem cells are plastic adherent fibroblast-like cells with the ability to self-renewal, that they can give rise to different cell types and tissues. These mesenchymal stem cells that we culture in our lab, which is CBCells, they exhibit a phenotypic and functional and oncogenic profile typical of mesenchymal stem cells, which proves that our cells meet the criteria to be used in cell therapy. And the last one is that these mesenchymal stem cells and their exosomes can be used as an alternative treatment for several conditions, including autism. And that's all. I don't know if you have any questions.
Yeah, I think so. First of all, regarding the oncogene, how do you make sure that the cells don't turn into cancer?
This is an important question. We check that these cells, they have these oncogenes intact, that there are no mutated, but basically this is going to depend also on the environment that they're going to be injected. If this person has some type of predisposition to develop cancer, it's better not to treat these people with mesenchymal stem cells, okay?
So
because
how do you check that the person doesn't have this kind of environment? What kind of test, a patient comes into the clinic, what kind of test does he need to do in order to make sure that treatment is safe for them?
It's going to depend on the doctor. That it's going to have their clinic history, basically, and it is very desirable that the doctor that is treating this patient check or like screen the patient to check whether he has like some type of predisposition to this cancer. It can be a family history of cancer, something that can tell us that maybe this person is prone to develop some type of cancer. But so far, the people that's been treated with these mesenchymal stem cells, they have not presented any development of cancer associated with the injection of mesenchymal stem cells. So far, the therapy with mesenchymal stem cells, it's been proved to be safe, but you need to have this in consideration to give this therapy to like a certain type of patients. You do have to analyze this and consider this with your physician when you want to inject like these mesenchymal stem cells in different patients.
It's just based on family history, or there are any tests to do that?
Yeah, like I said, it's gonna depend on the physician that is gonna apply this treatment to the patients. I would say that it might be based on the family history, but you can actually perform like a screening in the patients, but it's going to depend on the handling of the patient, depending on the physician that is treating this patient.
Okay.
Yeah, but one thing, Dr. Mariana, just one thing. In the certificate that the laboratory provides to us, it says that the donator for these stem cells, the woman that donated stem cells, she doesn't have any cancer in her family, right? And the oncological test, it says that these cells are not going to bring cancer to a new body, right?
Mm-hmm. Yeah, so let me add something. Thank you, Mariana, for your great presentation. We appreciate that. So let me tell you, maybe Hagai is asking about the markers of the patients. We don't need to do that because it's a little hard to get it and also expensive. It is not necessary because the stem cells don't promote any kind of cancer. Also, we just have to be clear about that the stem cells are not to treat cancer, and that's it. They don't promote it. Also, if some patients have a kind of cancer, we don't promote it with the stem cells, but it's not a treatment for that specifically. We use, for example, the Wharton jelly stem cells to recover the patients under oncotherapies like radio or chemotherapy. That is a great thing to recover the patient from that very aggressive treatments, but they don't attack the cancer. That's the thing. In some of the ways, the Wharton jelly were showing that they can have anti-tumoral effects and also attacks the metastasis. They can do it. So that's the reason that we use in cancer too. But none of the stem cells are indicated specifically versus cancer. That's the thing. They don't promote it, but they don't treat it. So We don't use to make the markers because they are a little hard to get it and expensive. We have to ask at least for 5 or 7 things of antigens. There are, for example, CA99, ACE, and also CA125. They're the most common. But we don't ask it because of that, because we have to be sure that the patient doesn't have a high or strong history of cancer in the family, and that's enough. Some of the parents were asking about the blood work for the kids, for example, some markers or exams, but we don't use it because we always try to do a comparison, a clinical comparison between the parameters. For example, in metabolic patients, we try to compare cholesterol, triglycerides, glucose, A1C, and we can see the improvements that. But in this kind of neuro things like ASD, it's so difficult to compare some parameters, some chemical parameters, because it's about behavior and conduct. So it's a clinical approach. That's the thing that we don't ask specifically for any kind of exams. If you want to do it, we could get it also here. We have the possibility to do it, but it's not necessary. So Mariana, I want to add something. And they were asking about the freeze or frozen cells. Could you clear the idea, please, about that? Because they are asking when we freeze it, are not freeze it, or is we have freeze it daily. So we are always repeating that we're producing fresh stem cells daily. So could you clear that, please?
Yeah, we do have our freshly isolated cells that we give into the doses that are going to be injected into the patients. The ones that you're getting in the doses, they are not frozen, but I want to clear something up. We do get cells at the beginning, and we're going to cryopreserve those ones because those are going to be the ones that we're going to keep like in a storage, okay? And from those ones, we're going to thaw them, and we're going to put them in culture to make them grow into the flask. And after they grow, we check them, and that's why we obtain them like freshly isolated.
Yeah, and very important thing that you say, Mariana, is cryopreserve, no frozen. Okay, this is very important, and this is what confused to all the patients. Cryopreserve.
And yes, they are cryopreserved. And it's important because it doesn't mean that you are going to take these cells and you're going to put them in the freezer and that's it. They have to have certain conditions that assure us that these cells, when they are frozen, they're going to remain viable, and that we're going to be able to use them like in a posterior time when we want to use them, and to put them in culture again. This doesn't mean that you're going to get the doses, and if you, like for example, if you didn't use them in these 3 days, you're going to be able to put it into the freezer, and I say, oh, okay, that's frozen, and I'm going to use them then, like later in time when I want to use them again. That's going to kill the cells. You cannot do that.
Yeah, I want to add something. Yes, with the cells, there are no frozen, but with the exosomes, we can froze the exosomes for 1 month. The patients can take exosomes with them, and they can froze just exosomes, no cells.
Yes, yes, exactly. And that's something that I didn't add in this presentation, and I meant to. The other advantage of these exosomes, we can storage them for longer than the cells. And as you said, Carol, yes, you can freeze this exosome and use them posteriorly when you need them. But because they are not like live cells, they are just a product of the cells that are releasing into the media. That's why you can do this, because it's not going to affect the integrity of these exosomes, but they do affect the integrity of live cells.
Okay,
also,
everyone
sorry.
has
sorry. So you have to be clear about the frozen one or the cryopreserved one belongs to P0. You know, is the zero
P0, yes, exactly,
yeah?
yes.
Because we have to start a whole culture with that one, with that kind of doses. There are the best one chosen from maybe 5 to 8 placentas, because of their characteristics. So we just want to be sure that you have to know that. We always observe the stem cells about the morphology and surface markers and everything. So we do all of the exams. They were asking Mariana because some of the viability tests that we given has 4 months before the administration. I think that we have to be clear that that kind of flow cytometry is performed by lots, you know, by the culture, whole culture. So we can repeat it in the lab. We do it also, I don't know if weekly or if 2 weeks, but we always do the cell counting daily and also the direct observation about the morphology.
Can you show the certificate, Mariana, please?
Yeah, sure.
They were asking about the date.
Yes. The date.
Okay, I know what you mean. It can be confusing because we produce the certificates, but a cell line, okay, for the ones that we are like producing. In this case, like it says L3001, okay? So at the beginning, when we like got these cells, and as Oliver is saying, Dr. Oliver is saying, we get the P0, which means the first passage of these cells, the ones or the first one that comes from the tissue that we're obtaining these mesenchymal stem cells. We put them in culture, we take the sample, it can be P0, P1, and we analyze them like through the time, it can be until the passage 15 or whatever you want, but we obtain the first ones and we make this assay, the viability assay for this cell line. We can start culturing this cell line in April, for example, but we have different cryopreserved samples from this same cell line. And when we don't have enough in the culture, we take that one and we start like culturing them again, so they're fresh again. See, I don't know if this like, I'm explaining this like clearly enough, but at the beginning we check this viability for the cell line and we check, oh, okay, these ones, they are viable for like 96 hours, okay, they're fine. They remain fine. We keep culturing them, etc. From like the passage 6, 8, whatever, and we obtain these doses from passage 3, passage 4, etc. But this certificate like belongs to a certain cell line that we already like test. We did the viability test, we did the flow cytometry, the oncogene, the microbiological analysis, and at the beginning is when we perform all of this analysis, okay?
Yeah, and also that the laboratory, you're talking about the passages. The persons need to understand that we have, the laboratory has a lot of passage 0. They took a little sample of passage 0 to do this test, but they still have passage 0 in order to give to the project, to Angels Project, to give us passage 1, passage 2, and passage 3. No more than passage 3. Yes. For the kids are just using these passages. It's okay, right?
Yeah, that's right. Maybe because I think that you asked these cells with these conditions and with these criteria, but yeah, it's exactly what we do. We just take the passage 0 to start growing them, and we culture them and we harvest them to give you in these doses up to the passage 3.
Yeah.
Okay.
And also, yeah, we have to let you know that there are lots of persons in Mexico producing stem cells with any kind of certifications, maybe going to 20 or 25 passage. And also there, some of them doesn't have any laboratories. So they are buying stem cells from a garage with no security or also kind of quality, but you have to be worried about that because the can put the life in danger, you know? That's a huge question.
So how can we know that we're choosing a lab or a clinic that is using a quality stem cells, real stem cells? what should we look for?
Well, the thing is that we are showing you that the certificates, the quality process. Also some of the people that are coming are able to go to the lab to know the installations, because there's a lot of people here selling maybe a little cheaper the stem cells or the treatments, and they don't have any kind of protocols of research or serious people working there. You have to take care about that because it's so dangerous.
Yeah, you need to know that the people working here in the lab, we are highly qualified people working in there in the culture area. That's why we are giving you this quality control certificate, which I don't think any other lab does, with all of these tests that we are performing and monitoring, showing you how we monitor these cells for cell line, and also we do it every day. And this is why I put lots of focus on how do we identify these mesenchymal stem cells, and why it is important to analyze these molecules to make sure or to make you feel sure and more safe that what you're getting are mesenchymal stem cells, because we have this analysis that we are performing in each of the cell lines that we are producing, which means that we have this written like support telling you this is the certificate that belongs to this cell, to belong to these doses, and this is how we're making sure that we are having our mesenchymal stem cells. We also perform the counting. We also perform all of these tests that, as I said, any other lab does, because we are doing it like every day and making sure that we don't have any contamination, that it has the best quality as you can get.
Okay, let's say I've decided I want to treat my child with autism with your products. How can I do that? How can I come to Mexico and decide that I want CBCells products for my child?
Okay, yes, the contact is going to be with me. I am Carol, with myself. You can contact me. We can arrange dates. You say, okay, I can go for this date. You will provide me the information about your kid. You will provide me full name, age, weight, allergies, diagnostic, and also you are going to provide me if there is cancer in your family. And then I will make a suggestion about the treatments that we can offer to you. One of these treatments, we are, I think, the only protocol that we are working with exosomes and with intrathecal sessions. The Dr. Oliver is going to explain about the treatment, but the intrathecal sessions are performed by a pediatric anesthesiologist. We are the only project that we have a pediatric anesthesiologist in our team. So then we talk about the doses, and we talk about the doses and the price and the dates. I block the date for 24 hours, no, for 48 hours. If you buy the tickets, the date is yours. If you don't buy tickets, I let the date open again in 48 hours.
Yeah, and also some of the parents can have some medical questions, and I can do the advisory of that, and we can talk about that and the treatments and the doses, because some of them are getting treatments too for the parents or for some relative that are coming with them, like grandmothers or grandfathers. They want to do something about other disorders, that we could do it. We could arrange the prices. We could also give you the same range of prices for other disorders that does not do it for anymore, because we are trying to treat the more persons that is possible. So we are giving great offers about that. And when we close the treatments, when we have to wait for you here. So we take like 2 days for the treatments. We always try to go to the hospital and take advantage of the sedation to do the IT plus the IV in the same time. And if the kids are able to be easy about the mask, we do the nebulizer the next day. Or also some parents are taking the options to get the nebulizer in the hotel and do it while they're sleeping. And that works good too, and plus the exosome with stem cells. or do it the same the next day in the clinic. And also we can program if you want or if necessary to go to the lab to know the installations and see what we are working on that.
Sorry, there's just one question regarding to the culture of the cells. And they're asking about the serum that we use. We use a fetal calf serum, which it is the main nutrient of the cells when they are in culture. This one is washed away thoroughly before putting the cells into a dose, okay? So you don't have to be worried that you have any debris of exactly FBS in the doses that you're getting, because we washed away thoroughly the cells before putting into the doses. Also, it is important to tell you that we are trying to migrate to a xeno-free culture, but we're working on it. But yeah, that's the question. That's the answer, sir.
They're asking if you can have a paper about a clinical trial with exosomes.
Yeah, there are some. Let me search for that, and I'm gonna share you. Yeah, we could find some. We have some of them. Also, okay. Plus stem cells too. Yeah.
Excellent. Now, how many people? How many children with autism, epilepsy? We have people with Parkinson's in our group.
Yeah.
Cerebral palsy. Do you have experience with all of these neurological conditions?
Yeah, absolutely. We are doing a very similar protocol of that, also in adults too, applying the IT, IV, and nebulizer, and we are having great results. So we have more experience about that. Also, sclerosis, multiple, and also lateral amyotrophic, other things that we are researching about. But we have experience in a lot of more conditions. Yeah, so we can talk about privately about a specific disorder, and we could give you a suggestion of treatments.
Okay.
Yeah, and also another thing, Dr. Oliver, that we work with patients with neurological disease. We work with Wharton jelly for the neurological disease. When the patients have diabetes or any lupus or any disease that is degenerative disease, we work with placenta cells. Mesenchymal, but placenta cells, and Wharton jelly are for the neurological, and also, like Dr. Oliver say, for recovery of cancer, not for treat cancer. For treat cancer, we have natural killer. Yeah. But for the recovery, we use Wharton jelly. This is important as well.
Yeah.
Okay, and I'd like your advice about searching for clinics in Europe, because many people find the travel from Israel to Mexico to be too long. You have to pass through Canada or the US in order to get into Mexico. There are very few clinics in Europe, but they are there. And personally, we've been to Russia and Serbia. We know about clinics in Georgia. Ukraine right now is not relevant, but we've seen it there. We've had a clinic from Poland on our group. So can you give us any tips how to figure out if these clinics are good? Maybe
actually
it's a little hard, but we could ask about a person of the lab, maybe Dr. Fabian, because the lab belongs to International Society of Stem Cell Therapies. And I think that we could find some info about the recommendations, specifically in Europe. Maybe we could help you some, but we are only sure about that we are doing, it's very hard to be sure of
no, of course, but what should I ask? What would I ask you to that I will know that you are a good clinic, that I can ask another place? What
Yeah,
should
as Mariana said about the quality controls, the personal, the protocols, kind of process that they are following up, how they're producing, if they're
The
for sale,
permits as well.
the
no?
parameters, yeah.
The permits that we have, like for the COFEPRIS as well, we have the license to culture the cells, that we work as a cell bank. And we have those ones that Carol can provide you as well. And the COFEPRIS is like the Mexican FDA. So we have the approval of them to do what we do.
We spoke to World Stem Cell Clinic in Cancun. They said that it's a problem to sell the products. They can't provide they have their own lab. They produce their own stem cells, and they don't sell the stem cells to other doctors in Mexico. They said that the laws in Mexico don't allow it. But I understand that your laboratory does sell to other clinics in Mexico. Is that correct?
Okay. Angels Hope is a project. Is separate to the laboratory. The laboratory is CBCells, and CBCells provide stem cells for all the country. The laboratory has the permission to provide the cells to all the country, but we work hand to hand with the laboratory, but we don't belong to the laboratory.
Mm-hmm.
And yes, there is permission to provide cells to.
Okay, so it is legal?
Yeah.
Yeah.
Of course. Okay. Does anybody in the crowd have any questions?
We talk about the neurological disease and
No.
I think that you're going to give a presentation now, no, Oliver?
Yeah, you
Okay.
want we could present about the protocol that we're doing.
Yes.
Okay.
Okay, if you don't have any more questions.
Mariana.
Thank you, Mariana.
No, thank
Thank
you.
you. Thank you very much.
Okay, and if you have any other questions regarding to the cell culture or the certificates, just tell Carol and I can give you the information that you asked for. Okay.
Thank you
so
very much.
Have a nice day and thank you.
You too.
Bye.
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 Thank you, Mariana. Carol, somebody's asking about Angel Hope certificate.
I don't know what it means. We work in a hospital. We perform in a hospital. There is a certificate of the hospital. I can give you the page. And we work with the certificate of the stem cells from CBCells. So we're a project, and we work with the license also. Dr. Oliver Leon, he has his own practice and he has his license in his office. So we work in the places where we have office. As Angels Hope, we don't have a license because we work under Dr. Oliver permission as a doctor, as his consultory.
Yeah, we don't need it. That's the answer. We don't need any kind of certificate particular about the initiative, the Angels Project, because we are. I have my own registration as doctor. We are working in certificate hospitals and certificate clinics, and under the permission of the lab. So that's it. That's enough. Regulatory, that's enough for working on that.
And our relationship with the laboratory, we've been working with the laboratory, myself, just for 4 years, Dr. Oliver for 10 years. He's trainer of CBCells. He trained a lot of clinics like Dream Body or a lot of clinics that you know. Dr. Oliver was a trainer, and he has a very strong relation with CBCells, but this is a project no under the name of
apart from the lab, but I work with them about the training other doctors and doing some actualizations about the therapies, also theoretical, practical, and doing some conference to patients too here in Mexico and also other countries in Latin America and also US. So we are doing advisory of every kind of treatments for the lab, but this time this kind of project is apart from the lab. So we are so close to them, but it's not a part of them. So we want to you to make sure about they are fresh cells produced every day, and that's why we are trying to get you here in Guadalajara, because we have to we want to get constant the criteria of the treatments and get the same kind of cell at the same time at the same place to make a comparison about the improvements. That's the way to do a kind of research of these or protocols. And we are to trying that to make constant the criteria to do every patient the same things. That's why we're asking to come here.
Yeah, and also we provide, Dr. Mariana talk a lot about the process and everything, but we are different also because we are the only project that when the kids are more hyperactive on way, the kids are not going to handle for the IV session, we can perform this IV session in the hospital under sedation. So sometimes the parents ask for this.
Yeah, we make it easy about that. Some parents are skipping the IT because some particular things. So we can perform the IV in the hospital under the sedations performed by Dr. Paola, who is a pediatric anesthesiologist with 20 years of experience. So she does kind of procedure every day. So that's are working great, and the parents are very confident about that, and that works great because it's so easy to every office. You know?
Yes, Dr. Oliver, if you think that IT has any benefit over IV. Yes.
Yeah, absolutely. Yeah, but the thing is that IT goes directly to the brain, to the nervous central system. That's the way to do it because we can skip the blood-brain barrier. That's a huge question because if we only use the IV, we could put some exosomes that can go through the brain. They can go to pass to the brain barrier, but the stem cells doesn't do it. So that's the way to go directly. We choose to suggest the 3 ways at the same time because we want to be very contundent, and we are seeing a lot of improvements, also immediately improvements about the kids. The parents are so happy about the results, and that's the way we do it. We suggest to do the 3 ways because we want to be stronger than the other ones. We have to be very contundent, and also we are offering great prices for that because we want to do that, to all the parents choose the way that we choose it and see the results very fast about that.
Yeah, another thing, Dr. Oliver, that we explain to the patients is that another projects, they give, like, for example, in Panama, the doses are very low doses. Yeah. And we decide to make doses like 5 or 6 millions per kilogram. Yeah. Because some of the cells, when you put in IV, some of the cells can stay in the lungs, for example, or in the liver, or in any organ that it has any problems. So not all the doses are going to be in the neurological system. And when you put it straight in the spine, it goes to the bone marrow, and it goes straight to the neurological system. That's why I think it's better the IT that over the IV, but together is excellent.
Yeah, the best way to do it is to do it together, you know. So let me go about the presentation if you want it, guys. So in some questions, search, we can find out how to answer. So we want to talk about stem cell therapies. We just have to confirm that the CBCells is the most recognized stem cell lab in Mexico. We have more than 14 years of experience and the highest quality standards that are not seen in anywhere in any more labs. So the labs count with two laboratories. There is the cell culture laboratory with all of the certifications that I'm not going to mention, but only we have to mention the COFEPRIS is the organ regulator like FDA in the US. Here is the COFEPRIS, that depends on the Ministry of Health in Mexico. And also a quality control lab that ensure and the quality and performs the test on the cellular products. So this is on the lab. We have 2 labs, and also we make a third-party lab, as Maria said, one of the government and one of private practice that are doing monitoring the quality of the products. So we always do that because we have to confirm that we are doing well things. About the process, we have to take the donor selections the first 3 months in the pregnancy. As Mariana said, we always choose between 3 donors, which is the best line, or 5 to 8 donors, sorry, and to choose the best line about the characteristics of stem cells. We do full placenta exams when we have it, and this is solved by Caesar surgery. We do the cell selection and expansion for cultures, and then the cultures and incubations. We do the microbiology test and cell countings, and we do the dose preparation daily fresh cells. No one does like that. The origin, we use 2 kinds of cell lines, as we said. We choose placenta because they have the best cell plasticity. As Carol said, to other disorders or diseases that are not neurodegenerative or cancer, we used to choose placenta because the plasticity is the ability to change their phenotype without genetic mutations in response to the environmental cause. For example, if the patients have diabetic, they go through the complications like the microvascular complications, and also to repair or regenerate the pancreas, etc., the liver. So they choose automatically how to work depending of the environmental course, or also the homing of the inflammatory markers of the patients. They are so specific that why we only choose in some disorders only to do the IV, because they work automatically. And also the Wharton jelly, because they have a great affinity in neurological tissues, and also, as we said, in cancer patients to get faster recovery and better. That's why we choose those 2 kind of cell lines or origins. About the regulations, as we said, we are regulated by GOFEPRIS and registered in the ISCT that I told you that is International Society of Cellular Therapies, and these are our license as cell bank. And we can show you it's registered officially in the government of Mexico. And also we are the only Latin American laboratory. No other one has this kind of research papers. We have this publication in a medical magazine performed in CBCells, and also Dr. Fabianario, it's on the authors of the research. We have a great result about this one. We do 2 protocols in the best research hospital in Mexico. There is a government one. It's called Salvador Subirán. It's at Mexico City, and we research 5 patients in worse conditions of COVID intubated, and we saved 3 from the 5. And then we repeat the model with 18 patients, and we saved 14 from the 18 only with stem cells. Well, about the ASD treatment with Angels Hope. This is an initiative that was created by Carol and me as a team. We are working together, and we take the benefits from the lab, doing a special line of stem cells for you, for the kids, and for the parents, and for the relatives. So who are we? We are a team of medical specialists that treats a long range of health problems with stem cell therapy, not only SDC, but this time is focused on that. We have 10 years of experience working with CBCells lab, and the stem cell therapies at the Angels Hope is an integral approach to treating autism spectrum disorder. So we create this for you, for your interest, and to take clear all of the thing with the treatment protocol. How does the stem cell therapy? Well, we can reverse the specific damage by administering multipotent cells that can stop the progression. We have to first stop the progression of the abnormalities and growth of behavior, and we can improve also abnormal behaviors like anxiety, hyperactivity, depression, and loneliness. And this is the thing about the researchers, that is by reducing the pro-inflammatory responses, and also about the immunomodulation the stem cells provides. That's the way we do it. What includes our protocol? Okay, we suggest to apply in 3 ways, as we told you. We start with the 50 million stem cells intrathecally in the spine. We have to add 5 to 7 million per kilogram in the IV, plus and dose of exosomes, and finish with 25 plus exosomes in the nebulizer to go inhaled therapy for the kids, and that works well. This kind of protocol we decide to establish this way because the impact study that are published in ASD and also neurological diseases, and also about our own experience. If you want to find some articles that talk about the nebulizer, it's so difficult to have it because there's none of information, but we are adding that because our own experience and the response of our patients in other kind of neuro things and also COVID or respiratory diseases. The nebulizer is a great therapy to complete the treatment. How is the procedure? Well, the IT application, as we told you, is done in OR and is performed by a pediatric anesthesiologist who is Dr. Paola Coelho. She's a pediatric anesthesiologist with 20 years of experience, and she was doing this kind of procedure every day from the last 20 years. So we do it under sedation by inhaling anesthetic gas. There is sevoflurane or deflurane. This is diluted in oxygen because we need to keep the kids easy to avoid the pain and moves that can hurt them. So it's too easy to do it. They go to sleep like 20 or 30 minutes. Only the gas. We don't use IV things or IV medication because that can be having lost the effects of the anesthesia during the day. So they wake up after the 30 or 20 minutes and they goes very well. So they can go and eat something and you can go to the hotel, back to the hotel. And also we take advantage, as we told you, of the sedation to do the IV applications, or some parents that are skipping the IT way, we could do only the IV in the hospital. And we have to only charge about the hospital expense and the anesthesiologist. That's the way we do it. Then we try to do the nebulizer the second day of stance here in Mexico, because we want to follow up the treatments and see how the kids are reacting. And if they can keep the mask, we have to do it in the clinic. But some parents are asking to do it in the hospital also, or while they are sleeping in the hotel, we can give you the advisory how to do it. It's so easy to do it, and they can lie while sleeping. Does stem cell therapy has the same result for everyone? No, it's depending of several factors like the patient's health condition, underlying issues, lifestyle, and other. But we always have gains, absolutely. The improvements are very variable, but we always getting better, all of the patients. That's the thing that we have to value about this kind of disorders. We have to value the behavior and the conduct of the patients. What are expected about the results of Sct? Well, according to our results and clinical outcomes and pre and post treatment observations, we can show to help in reducing the progression. That's the more important. Yeah, you have to stop the progression, and that's a huge gain. But we have to speed up the tissue repair and also regrow and repair tissues after 6 months. We always have a chance to be getting better the conditions, also the conduits, and they are showing that, but we have to value with the days going on. The result one, well, we can see after the single infusion, and you can expect a significant improvement in the behavior and conduct of a child. We have more independence, and IDLS and IADLS is like the daily task conducts, mobility and coordination, and gait after 6 months of injection. They are showing very early about that. So we have to see some improved also the first week of the treatment. So that's we are seeing in the patients. Also, some of them are going back to school after some years that they cannot go in. And the teachers are very surprised about the behavior of the kids. And also we could increase the independence, more life satisfaction, decrease damage of progression, and reduce memory decline. That's the most common effects that are reported. And also we are seeing that in our own patients that we treat with Angels Hope. This is the whole abstract of the treatments that we are doing here. We hope that we are being very clear about this. And I don't know if you can have some questions or something.
Dr. Oliver, yes, actually, so someone asked me yesterday about this that there is any explanation about the kids have a temporal regression.
Yeah, so we come, the first thing that we do with the stem cells is to stop the progression. And then we can start we'll start to see the improvements. It's that gradual, and we have to make a daily observation about the behavior of the kids, but they are doing very well about that.
Hi, I'm Sheila. I'm Chagai's wife. We spoke
Hello.
earlier on the phone.
Yeah.
I'm also a nurse here in Israel.
Yeah.
I have 3 children with the same condition.
Yeah.
So they have epilepsy, autism, and my oldest, he also has a disease called EOE. So esophageal, well, I'm very tired, I'm sorry. He has eosinophilic esophageal disease. And we are not having any success right now. He's done 4 rotations of stem cells.
Right.
And he's due to do his 5th. And he's only gotten IV and exosomes in the past.
Mm-hmm.
But his stomach is not doing very well. And we are wondering if there's anything that you think that stem cells can do for his digestive system that is very damaged at the moment.
Okay. Yeah, I think the way to go about that protocol in specific is to add at first the IT application to see what about the improvements of that way. I don't know which doses you have, but
he received 2 million cells a kilo.
okay. Okay, so
How many kilos he is?
He's done 4 rotations. 3 rotations in 1 year, and then there was COVID, so he got nothing for 1.5 years. Mm-hmm. And he's done 1 last summer, and he has another one to do in about 3 months. He got 2 million cells a kilo per injection, and he's gotten so far 7 injections.
7 injections. Okay.
Yes.
So how many kilos is the weight?
Right now he weighs 26 kilo.
And he's getting like 50 each time.
Yes.
Okay. Yeah, so maybe we have to try our kind of protocols to get up a little bit the dose and also apply the IT way to look for improvements. I don't know.
When we tried in one of the rotations, we tried to give him an injection that was 3 million cells a kilo, and he had a very bad response. So he was shaking, he was shivering, he had a very high fever. He was obviously right away treated.
Do you know the origin of those cells?
Yes, he was treated by Swiss Medica in Moscow in that particular rotation. And it was placenta, and I think that one was umbilical cord. The second injection that he got when he got in jelly stem cells. But I think it has something to do more with the doses, because he got 3 million cells a kilo on that this one time. And it wasn't good for him. He didn't respond very well. He did respond So he was in diapers and using baby formula at nearly 6 years old when we did the first treatment, and had very severe epilepsy, which is now for the past 3 years since the first treatment completely gone. Excellent. His EEG is normal.
Yeah.
And he started speaking, he got weaned off of diapers, he eats regularly, and he's able to communicate pretty well what he wants to say. He's not normal in his communication abilities, but he's doing a lot better.
Wow.
However, we've been struggling with EOE in the past year. We just didn't know before. He wasn't able to communicate enough to tell us that he's sick. But once he was able to explain that he's not feeling well, we were able to start looking into what is wrong. And we found that he has EOE. And we've been doing very poorly in the last few months, to the state that he has gastritis right now. Is there anything that you think that we can do to improve the situation?
Yeah, so, but we suggest to complete the protocol as we are doing and see what about the results. That's the only way to do it, to compare about the ways and the doses. I think that's the best way to treat them, you know. It's we don't have anything for sure, but we are having improvements in all of the patients at different levels, obviously, but we have to try it. That's it.
How many treatments do you do in your clinic? We've done 4 for this child. So double injections 4 times, where he got for each injection 2 million cells of keto. And in the clinic we are treated in, we were told that 5 rotations is the max. They don't do more. How many rotation is from your protocol acceptable?
Yeah, we are suggesting to repeat it 3 times at least in a gap from 2 to 4 months, maximum 6 months each space. That's the way we are doing that. But we choose to apply the 3 ways at the same time to absolutely see the improvements. So that's our parameter to determine if we are doing well with that.
So if
and there is no maxim, there is no maxim about the rounds. Like you say, there is no maxim. For example, if you apply 3 times per year or 4 times per year, there is not any problem if you are going to treat maybe next year or 2 years later, there is no problem. With the intrathecal, this application, it has to be done 3 times at maximum, and then you can start just with the maintenance with just IV sessions. But there is not a maximum even in adults or in kids that about stem cells.
There are no risks about overdoses or something.
So I can continue giving him stem cells if necessary for years to come.
Yeah.
Yes. Also for adults, for example, for adults, when they have a chronic disease or something, the stem cells are not going to heal the disease, but they give expectative of life. So a lot of adults, they put cells every 2 months for years.
If an adult, a person had prostate cancer, it was treated, and then he wants to do a stem cell therapy, is that allowed?
If he's cancer free.
We could do it. We could do it, but we have to be sure that it's on remission with a negative study.
It's post radical prostatectomy.
That's it. Let's do it. Yeah.
Okay. Another question, and I'm asking for a family member. I have a family member with a history of Alzheimer's. They have Alzheimer's disease in their family, but they also have like muscular spasm in sleep.
Yeah.
And diabetes type 2. And I'm wondering if this kind of treatment can be beneficial for them.
Yeah, absolutely. We could avoid to present the progression of that kind of diseases that we are proclive to present about the heritage or the parents with the same. We have to do it because we have to maintain the quality of life and also get better about the metabolic disease that is diabetic. We can assure that the patient doesn't gonna present the complications of diabetics, and also we made some adjustment to the basic treatments like the pills or insulin, that kind of stuff.
And for the Alzheimer is when it's in the first stage of the Alzheimer. When it's very advanced the Alzheimer, there is not going to be any gain in the
Yes, I know.
Yeah.
Can you talk a little bit? I don't know if we spoke about it because I was busy with my baby who's also autistic and epileptic. As I said, I have 3 of those.
What's the youngest age you can come?
The youngest?
Yes, we have a 1 year and 4 months old baby. He's unstable with epilepsy medicine. He's now being diagnosed with autism. Wow. It's the same genetic condition like his siblings do. And they responded very well to stem cells and cord blood. And we are wondering how soon can we treat him
Yeah, we have I think the youngest one was 2 years, Carla?
He's 1 and 4 months right now, our son.
Yeah, we have to figure about that with the anesthesiologist because I think that the youngest one was 2 years, Carla. Am I correct? 2 years or 2 years and a half, I think.
2 years, 2
Yeah.
years and a half.
Yeah.
2 years and a half because it's when the autism.
We have
is
the
like
diagnosis, definitive
yes.
diagnosis. Yeah.
Oh no, this baby has autism. It's he's nonverbal, completely nonverbal. He doesn't sleep. He has very severe epilepsy. We know. I mean, it's the same genetic condition. Another question, I'm not sure if you spoke about this, but we have a lot of people in our group that has teenager children with autism who's not done their first treatment. And when they go to different clinics, they say that the treatment is not so effective for them. What do you think about it?
We don't have any kind of problem like that. We always have improvements. As I told you, at different levels, but they always have gains because that's the reason that we choose to do the 3 ways at the same time. We never had that.
So if they don't want to do intrathecal, because the doctors here in Israel are very against intrathecal.
Yeah.
And I have to say, as a nurse, I have done many intrathecal procedures on children. I was also an OR nurse for many years, and there was always a problem. They always had difficult time to recuperate, or they weren't very compliant with lying down afterwards, and it's painful. Intrathecal.
No, the intrathecal session, when the anesthesiologist perform the intrathecal session, the kids are totally asleep. So they
yes, but it's always there. They are sore after. So even in Israel, we do intrathecal while the children are sedated.
Yes.
And a lot of the times I saw the children afterwards and they had some pain. And it was treated, and you know, it's normal if somebody stabs you in the back, it can be painful.
Yeah.
Even if you were sedated. The other concern that we have is especially because these children, they're being treated in a foreign country. And very soon after the intrathecal procedure, they will go on a plane and if anything goes wrong, if they have a CSF leakage in those 72 hours, which can of course happen after intrathecal, it's normal complication.
But after too many hours, no. Yes. We always do this kind of treatment because you have to know as personnel of health that this kind of effects have to be presented in the first hours. That's it.
It doesn't always present in the first hours. Sometimes it can be presented after 24 hours. But we don't know if the patient
that's why we ask you to keep 2 days here, and that's enough for you.
So you have a minimum of stay for 2 days.
Only.
Yes. Yeah. And if they need a blood patch procedure, you can perform the blood patch procedure.
We never need that because we use a very little needle, and the doctor is very capable to do it. She's always doing that from the last 20 years. We never had any kind of complication.
Okay, but if there will be a complication and the patient will need blood patch, you are able to perform.
Yeah.
Yes, it's performed in a hospital, in a OR. Everything is like, todo está preparado. Everything is prepared in case of an emergency.
If you speak in Spanish, I understand.
No, yeah, but everything is prepared for a case of an emergency because we perform in a hospital, not in a clinic, in a totally well-established hospital. And we can perform everything in one session. We can perform IV, nebulization, and IT in one session, but we ask for the nebulization at the same day in order to see the kids, in order to second consultation. Some kids, even without IT session, they can have a fever. It's totally normal. If they can have a fever in the night, it's totally normal. We say that they can bring Tylenol or Neumelobrin or ibuprofen. The thing where they are not allergics or something, they can give in the night and a shower. And in the morning, Dr. Oliver see the kids. If everything is okay, they can leave. So that's one thing that I was going to say. And the other thing, when you say that there is a lot of parents that they didn't see any gains, first thing, we talk about the cells from CBCells. We can see that CBCell cells are working, first thing. And the second thing, we see not just kids, we can see adults, persons with diabetes, everything. It's not like a kitchen recipe, a cooking recipe. You know, it's like a lot of persons react better. They have more gains than another persons. It is not a rule. It's not like a, how to say, like a cooking recipe, you like know, that you put 2 eggs and you put this and you're going to have a cake. It's not. It's something that we're working. Is every person is different. We have a protocol, a similar protocol, and the kids are going to, their bodies are going to say how many sessions they will need.
Mm-hmm. When do you say to a family that enough is enough? So this family has been coming again and again and again. When do you think is the time for a family to stop doing stem cells?
When the family decides.
Yeah, I have to decide it because of the behavior and the evaluation of the doctors in your country. That's the way to do it. We have to that's why we are suggesting to repeat it 3 times, because it's enough for almost all of the cases, but they have to see what about the specific response of the kids. We cannot decide a specific time for all, because as Carlos said, everyone is different, so.
Okay. I'm sorry if I'm asking difficult questions. I feel sometimes that this is my job.
Yes,
also because me and Kagay are directing this community and we assist people to arrive to clinic like yours, but also because I think that there are very little opportunities for the Israeli crowd to hear medical professions from other countries. And I find it very important for the people to know that it's okay to ask these kind of questions. I am wondering, what is your opinion about cord blood as a stem cell procedure? Is it better? Is it not as good? Because this is a question I'm being asked very often.
Yeah, that can be used too, but we choose that kind of origins, that is placenta and Western jelly, because that, as we told you, it's the best to work. We have to capable in the lab to produce any kind of stem cells, whatever we want. But we choose the both of the placenta and Western jelly because that questions. We have to use the best plasticity and also the more affinity of the neurological tissues. That's why we choose that. That doesn't mean that any kind of stem cells are not good for treatments, but we choose that because our own results and experience.
Yes, and also I heard Dr. Fabian, the owner of the laboratory, talk about the cordon blood are better in leukemia, in cancer, or just a few disease of the kids. So a lot of parents here in Mexico as well, they keep the cordon blood, but it's just going to be used in any case of leukemia or any case, a few cases.
And in a blood disorders. More related.
Our son, one of our children, as I said, we have 3. Our middle child received cord blood, and it was a very successful procedure for him.
Yeah.
He received it through the IV, and we are considering to take him again this summer to do a stem cell procedure just to complete the treatment. So I'm just wondering, because right now, when we tell young parents who are about to have their first child, we always explain that they should preserve cord blood. And I'm wondering together with you if this is a right instruction, if we should continue telling parents to preserve cord blood.
Well, here in Mexico, we don't recommend that because it's a unnecessary expense, because there's some other labs that are producing every kind of stem cells. So also it's more expensive to process the cord and to do the process, the maintenance, the cryopreservation. You have to spend more than only apply stem cells. Here in Mexico, that's not
The
practiced.
question is, it's a matter of cost, because here in Israel,
not about the cost.
It's a lot cheaper.
It's not necessary. Not about the cost. It's not necessary. That's it. We could get stem cells wherever you want it, whenever you want it, and all of the regions that you want it here in Mexico. That's the thing. So it's not necessary to spend to have that kind of benefits.
Okay. Dr. Ortiz from a different clinic in Mexico was giving a lecture here before, and he spoke about stem cells being very bad travelers. So they don't travel very well. What is your opinion about this? Because I know that CBCell sells your cell to other companies as well, other clinics as well. We've discussed it in the past. And even though you manufacture very good cells, the question is, how well does your cell travel to other clinics?
that was great. We have performed some tests in other sites from here in Mexico, and also the first one, that is Tijuana or Merida, and we have great viability and also the conditions of the cells. We perform that kind of exams and everything works well. So obviously the thing is that viability goes down a little bit. That's why we always put like a 20% more of the doses that you are buying to cover that kind of loss about the time. But we can assure the 3 days viability in all of the doses that we send to other places. That
from the moment they leave your clinic,
it
is good to give for 3 days?
3 days only, that's it.
Okay, so if I want to know that the clinic I went to and use a CBCell cells, if they are good, is there like an expiration date on the vial? Yes, and I can look as a patient, look at the vial, and it will say how many cells are on it, and the date of expiration, the date of manufacture. And any patient that goes can see the vial and say, okay, this is good.
Mariana explains all of that in the first presentation. Everything was very clear about that. So we always do that.
The time is almost over.
Okay. Thank you very much. That's all my questions. If anybody else would like to ask, you're more than welcome to.
Okay, some of the parents were asking about the regression that they present. So we don't have any kinds of, they present that. They always were improving and they don't have any kind of regression. I don't know what exactly you refer to, but we never had that kind of problem here with our kind of cells and our protocols. Okay, and also about the to not get benefits. There are some exclusions about the protocols. Some of genetic diseases are not too able to improve. We could stop the progression, and also we have more benefits in other sides of the body. But with something that are genetic determined, it's very difficult to get done. But we always have improvements in all of the conditions. I don't know if you have more questions or something.
Did you see the list of questions on the chat?
We'll respond all of them, and I was finishing that.
If anybody has any more questions, you're welcome to ask. If not, I think that we are finished for tonight.
Okay. Thank you. Very, very nice to meet you by this way, and we are here to help you, to serve you, and we hope to see you soon here in Mexico. Thank you.
Thank you very much.
Thank you very much. If you can send us the recording so we can upload it to the group, and parents who are not able to join us, or future parents who are not yet in our group and would like to hear you can hear it, will be very good.
I will send it.
Very, very much. Have a lovely evening.
You too. Thank you very much. Bye-bye.